Okay, now on to non-competitive inhibition. Write in your notes that in non-competitive inhibition, the inhibiting compound does not compete with the natural substrate for the active site on the enzyme like a competitive inhibitor does. But it inhibits the reaction by combining with the enzyme substrate complex after the complex is formed, and then by doing this causes the actual enzyme to change shape. Also write that this is referred to as a conformational change, and that this type of inhibitor is called an allosteric inhibitor. And then the site that binds to it is referred to as the allosteric site. So let's go over that in this diagram a little bit. So here you see a typical enzyme substrate complex. And then you have the allosteric site and the allosteric inhibitor. Now, once that allosteric inhibitor gets into this allosteric site, so it fits, there's a conformation there as well. It changes the shape of the enzyme, and you can see that here. Changes the shape of the enzyme so the substrate can't fit in. Now, remember, a competitive inhibitor competes for the actual binding site. So once it comes in to the binding site, then the substrate can't bind to it. Now, in allosteric inhibition, we have an allosteric site or someplace other than the binding site on the enzyme, and that molecule then lodges itself in there and then causes the shape of the binding site to change, and therefore the substrate can't bind to it. That was a question on the 2013 exam. So make sure you have that straight. You'll be seeing that again. So write in your notes that once the non-competitive inhibitor is bound to the enzyme, like you see here on the bottom right, that bond changes the shape of the enzyme. And that when this happens, the active site is no longer a functional binding site, which keeps the enzyme inactive. Also get in your notes that enzyme inhibition is the basis for most chemotherapy treatments, and that many health disorders can be controlled in principle by inhibiting selected enzymes. And then an example would be a chemotherapy drug called methotrexate, methotrexate, which is a common anti-cancer drug. Now, this works by inhibiting the enzymes involved in the synthesis of thymine and hence DNA. Now, as a result, this stops the production of more cancer cells. Cancer is simply an out-of-control cell growth. Something happens in the cell cycle, which we're going to learn about here in a little bit. Something happens in the cell cycle, and the cell cycle doesn't know when to stop. So it keeps producing these cancer cells like crazy, especially if it's a viral cancer, out of control. And that out-of-control cell growth would form a tumor. Well, methotrexate interrupts that now by changing the enzyme, all right, that allows for these cancer cells to continue to be produced and stops their cancer growth. Now, another example would be cyanide poisoning, which is an allosteric inhibitor that stops the production of ATP in the cellular respiration process. So my point is, there are two examples you need to have for non-competitive inhibitors. Both work the same way, but if they ask for more than one example, you now have more than one. Okay, let's move on now to irreversible inhibition. Irreversible inhibition. Write that most inhibitors are temporary, meaning that they are reversible. That there's a way to reverse them or get the inhibitor off of either the active site or the allosteric binding site. And then in an irreversible inhibition, An inhibitor, once bound to a protein, cannot dissociate or become unbound. Also write that in irreversible inhibition, the inhibitor is permanently bound to either the normal or allosteric binding site. Very important that in your mind you get a clear picture of the difference between the normal binding site and the allosteric site, so that once you see the word allosteric inhibitor, you understand the mechanism involved. Now, a good example is provided by the nerve gas DFP, which is designed for use in warfare. So we want to start with that example. DFP, it's got a big long name, but it really doesn't matter. You can use DFP on the exam. Write in your notes that DFP combines with the amino acid serine, S-E-R-I-N-E, at the active site of the enzyme, and we're going to see this enzyme again later, acetylcholinesterase, A-C-E-T-Y-L. Is it on here? Acetylcholinesterase. There's cholinesterase right here. A-C-E-T-Y-L. So there's the acetyl, and there's cholinesterase. So acetylcholinesterase. So this DFP gas combines with the amino acid serine at the active site of the enzyme acetylcholinesterase. Now, the enzyme deactivates the neurotransmitter acetylcholine, and you see that down here. Now, neurotransmitters are needed to continue the passage of nerve impulses from one neuron to another across the synapse. Most of you probably have anatomy and physiology. We can think of nerves as consecutive brooms. If you think of a regular broom type thing, the stem is the axon, and the broom part now would be the neurotransmitter or the dendrite, and there'd be a space between the brushes of the brooms. Well, nerve impulses jump across that space when they're transmitted from the brain across your body. And these neurotransmitters are needed to continue that passage of nerve impulses from one neuron to another. Now, once the impulse has been transmitted, the enzyme acetylcholinesterase deactivates the acetylcholine almost immediately by breaking it down. So make sure you get that in your notes. So this is acetylcholinesterase breaks down acetylcholine almost immediately. Now, let's talk about the DFP gas. If that enzyme is inhibited, acetylcholine accumulates in the body and nerve impulses can't be stopped, causing a prolonged muscle contraction. If you know what a cramp is like, all right, typically now cramps are stopped by the release of acetylcholinesterase. Well, if that acetylcholinesterase is blocked, there's no way to stop that cramp. And what happens is paralysis occurs, and death may result since the respiratory muscles are affected. Make sure you get that in your notes. Now, the muscle responsible for respiration is your diaphragm. So if that diaphragm is stuck in this cramp, then you can no longer breathe. Well, if there's no way to stop the cramp, then obviously no breathing is bad. So you can die from that. Now, also write that some insecticides currently in use, including those known as organophosphates, write that down, organophosphates, and I want you to write an example, parathion, P-A-R-A-T-H-I-O-N. So those known as organophosphates have a similar effect on insects. It can also cause harm to the nervous and muscular systems of humans who are overexposed to them. So if you're using now something for ants, like there's an anti-ant insecticide called malathion. And malathion does the same thing. Well, you got to watch it. If you have pets or kids, if you use too much malathion in places they can get to, it can have a bad result. Okay, let's move on to something called allosteric regulation. Anytime you see the word allosteric, you're going to think of that site on the enzyme other than the binding site, right? So when we look at these diagrams that I have in this slide, you can see now the active sites right here. These are the normal active sites on this particular enzyme. And you can see now that this activator is in an allosteric site. Okay? So here's the inactive form, here's the active form. So we're going to talk about something called allosteric regulation. Now write in your notes that allosteric regulation is the regulation of an enzyme or other protein by binding an effector, E-F-F-E-C-T-O-R, effector molecule, by binding an effector molecule at the protein's allosteric site. That is a site other than the protein's active site. So if we take a look at the diagram in the slide, we can see where the binding site is located. Again, we already kind of briefly went, there's our binding site. And also where the allosteric site is located. So here's our allosteric site right here. Also write that effectors that enhance the protein's activity are referred to as allosteric activators. Allosteric activators. Those are ones that enhance the protein's activity. Whereas those that decrease the protein's activity are called allosteric inhibitors, which you see right here. Now the difference is activators now would keep the enzyme in its active form, whereas inhibitors then would keep the enzyme in its inactive form. So get that down as well. The term allostery, where the root for allosteric is, comes from the Greek allos, which means other, and stereos, which means solid, in reference to the fact that the regulatory site of an allosteric protein is physically distinct from its active site. I don't know if they'll ask that on the test. I've never seen it, but it never hurts to have that information. Allosteric regulations are natural examples of control loops, such as feedback from downstream products or feed forward from upstream. More about feedback in a couple of slides. Okay, so let's try and organize what we've learned about enzymes into some logical sequence that you can see and use. Now, we're going to divide this up into steps, and we're going to number the steps. So let's talk about step 1 first. So you're writing now. In step 1, write that the chemical reactions of metabolism are organized into what we call metabolic pathways. And metabolic pathways are one in which a chemical is transformed into another by a sequence of enzymes. And you see that right here. This is a metabolic pathway. And you see there's more than one enzyme in this pathway. Okay? So each of these enzymes now will produce a product that will contribute now to the next step, and so on and so forth. So the enzyme here produces B, B then contributes to C, C contributes to D, etc., and the final product now would be G. In step 2, write that enzymes are crucial to metabolism because they allow organisms to drive desirable, that means that's good, but thermodynamically, energy, unfavorable reactions by coupling them to favorable ones. Okay? So step 2, enzymes are crucial to metabolism because they allow organisms to drive desirable but energetically or thermodynamically unfavorable reactions by coupling them to favorable ones. Okay, in step 3. Step 3, write that enzymes are crucial because they act as catalysts to allow these reactions to proceed quickly and efficiently. Boy, do you know that for sure. Step 4. In step 4, write that enzymes also allow the regulation of metabolic pathways in response to changes in the cell's environment or signals from other cells. And what you know about signals from other cells. Now, in step 5, write that it's the metabolism of an organism that determines which substances it will find nutritious and which it will find poisonous. For example, some prokaryotes use hydrogen sulfide as a nutrient, yet this gas is poisonous to animals like us. Step 6. In step 6, write that the speed of metabolism, also called the metabolic rate, also influences how much food an organism will require, and that a striking feature of metabolism is the similarity of the basic metabolic pathways between even vastly different species. For example, we're going to talk about the Krebs cycle, also known as the citric acid cycle, when we talk about cellular respiration. This is present in all organisms, being found in species as diverse as bacteria and huge multicellular organisms like elephants. Now, and lastly, the last step, write that these striking similarities in metabolism are most likely the result of the high efficiency of these pathways and of their early appearance in evolutionary history. Now, I know I went a little long on this one, but that last explanation is important. So that's done. We're done with part 6. Take a picture of your notes, submit them to Moodle, grab that part 6 study guide, and I'll see you in the last part, part 7. Bye.